Hello, 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 and welcome to this third screencast on the cell introduction to cell biology test. Today we're going to be talking about meiosis, and we're going to be preparing you for the third question in the test. Now, the first part of the question asks you to name a process. Now, naming something is very easy. It's just a name, and that's all you need to do in order to get through the test. What I'm going to hopefully be showing you is how you can build an answer to have that more depth and more quality to your answer. But don't be afraid just to write one thing. If you're struggling, if you are struggling for time, effort, etc., you're struggling with the kids, struggling with illness, whatever, then simply a word will do. Now, the second part of the question, traditionally over the past 7 years that I've taught this, seems to be the place where most students struggle. And basically what we're going to be doing is outlining the stages of meiosis. So it's basically about how the chromosomes move to cause very special things in the human body. So hopefully I can simplify that for you, and the whole point of doing this screencast is targeted at that second question to try and make it as easy and as well understood as possible. The third part of the question is also quite simple in that you're explaining the biological importance of meiosis. So it's a three-pronged question. The second part of the question is the most difficult, which is where we're going to focus most of our attention today. Okay, let's start with the first part of the third question, in which you're asked to name the process in animals which requires meiosis to take place. And like I said in the first slide, is that really you're looking for one word, and hopefully the better answers will be put into a sentence. And the process which requires meiosis to take place is something called gametogenesis. Sounds very posh, and I hopefully have written it clearly enough that you can spell it Gametogenesis, gametogenesis. And to break the word down, a gamete is a sex cell, which could be sperm or egg, spermatocytes or oocytes for eggs, and genesis means creation in the biblical sense. So the process in animals which requires meiosis to take place is called gametogenesis, which is the production of sex cells, i.e. eggs and sperm. And that's really all you've got to write. That is the answer for the first part of the question. Now, even if you put the creation of sex cells, at least that's an answer, and you can get away with the answer by doing that. But if you mention those terms right there that you see on the slide, you are going to answer the question nice and fully. Now, if you ask the question, why do we create sperm and egg cells? Why is it different from creating any other cells within our body? Well, for this we have to kind of look back to mitosis and give a comparison, because mitosis was the exact copying of the chromosomes. So mitosis, we started with a cell with 46 chromosomes, and we ended up with two identical daughter cells with 46 chromosomes. Now, what is different is that sex cells, i.e. sperm and egg cells, have 23 chromosomes in them, which is a key difference of meiosis. Meiosis is about taking your chromosomes and creating sex cells that have half the number of chromosomes than your usual cell. So this is the full complement, i.e. 46, and we would describe that as being diploid. Diploid means full complement. half the number is termed haploid. Haploid. Or halfloid, if you like. Now every creature has its own diploid and haploid numbers. Now I'm going to make these up because I don't have them to hand, but let's say a dog, a border collie, has 112 chromosomes being diploid, and in its sex cells obviously that would have 56 chromosomes in its sex cells, and so on and so forth for the different species such as fish and chimps, etc, etc. But all diploid means is a full complement for that species, and a haploid means half the number. So to recap, when you're answering this question in the test, name the process in animals which requires meiosis to take place. Simply put, the process is called gametogenesis. This is the production of sex cells, i.e. sperm and egg. If you want to go into the numbers, it's about producing haploid cells from diploid cells. You can do. But what we need to do in the next couple of slides is to give you how that happens and why it happens. So we're now going to zoom to the third part of the question, because I feel like it moves on naturally from there, is explain the biological importance of meiosis. Why is it important? Well, again, if we compare it to mitosis, mitosis is an exact copy. And the importance of that is that we're able to create cells that are able to do the same jobs that we want them for. So if our stomach lining broke down, we could repair them. If we want to grow a particular part of the body from being young, then we can create new cells that have an exact copy. But meiosis is completely different. And like I said in the last slide, it's about creating gametes or sex cells. So what's different about sex cells? Well, this relates to the reason why you do not look exactly like your parents, because meiosis is about us taking one of our cells, so let's say this cell here, which could be in the ovaries or it could be in the testicles. What a great thought. And when we want to create sex cells, each cell then goes through two different splits. So the first split means that we take these 46 and we split them in two. And actually what happens is these get split in two again. Now I'm hoping you're going to think in your head that doesn't make sense. Well, in the number form, if you just saw it like that, it probably wouldn't make sense. Because how does 23 split into two 23s? Keep that question in your mind, because it's a key question and it will help you explain how meiosis is important and why meiosis is important. But that by the time we create sperm or egg, we have randomly assorted, we have genetically sort of selected some chromosomes to go this way and some chromosomes to go that way. So this cell here will be completely different to this cell here because it'll have a different component of the 46. It's like taking 46 playing cards, shuffling them up into 4 different piles, and you end up with different chromosomes. So to answer the question, explain the biological importance of meiosis. The first importance is for genetic variation. And if you want, you can just write genetic variation in your answer. Okay? You can just write that and you will get the pass. I'd much prefer it in a sentence explaining, but that's going to just about get you through the exam. The reason why meiosis is important is because it gives us cells that have a varied genetic makeup. The other reason, which links from genetic variation, is that we are able to evolve, or evolution. Over a period of time, these genes, through generations and generations, allow us, because we pass those genes on, to evolve and to adapt to our surroundings and to our environment, which allows our species to be more robust in terms of survival. The third reason why meiosis is important is because of the mathematics involved. We take a cell of 46 and we create cells with 23. So that means when a sperm meets the egg, we create another cell of 46. And so the cycle keeps going. If the sperm had 46 and the egg had 46, we'd obviously have 92. And then if the next sperm had 92 and 92, it would just go up exponentially. So one of the third main reasons why meiosis is important is to maintain chromosome number. So for every generation, we split our genes in half. 23 chromosomes come from the sperm, 23 chromosomes come from the egg, and hence a new human is being created. And then when that human gets either to an adult, a boy or a girl, and they start creating sex cells, then their sex cells will have 23, and they'll meet with another boy or girl to make 46, and so on, and so that chromosome number is maintained throughout the generations. Okay, so that's an overview of what you need to write in the third part of the question. If you just want to write these 3 bullet points, that would be satisfactory. If you can explain them in a sentence, then great. The meiosis is important for genetic variation, so it creates cells with a different genetic makeup each time, which then allows us to evolve as a species over generations and generations. And the fact that we take a cell of 46 chromosomes and make cells with 23 chromosomes in allows us to maintain chromosome number. Okay, that's the answer for part 3. So all that's left is to explain the key chromosome movements, and the questions in your mind should be: how do we get a genetic variance? How does this split or these splits cause us to achieve 4 different cells that have different genetic material? And that's what the next slide will be on about. Okay then, without further ado, what we're going to look at is the second part of the third question, which historically has been the trickiest one for me to teach and also for learners to learn. There's quite a bit of terminology which I've tried to help you with the word wall down the right hand side here. So if you're struggling with a term that I use, rather than me go through it, then pause the video, have a look at the word, get it in your head, and then carry on with the video. I'd much prefer to do it like that, because otherwise I've already done about 6 takes of this already and it gets a little bit messy. I'm going to go for cut to the chase, what you really need to know, and you use this document and video to the best of your ability to help you piece together the different bits. Okay, so there are 2 aspects that you need to know about meiosis. There's the numbers, so how many chromosomes are involved at each stage, and then there is how do we get genetic variants, or how do we get different genes when we're creating sperm and egg. So let's take the numbers first of all. Numbers. Now the numbers are represented at the top here. 2n at the top meaning diploid, meaning 46, and the n's mean 23. Now when we look at this, as I said in the previous videos, how does 123 split into 2 23s? And the answer lies in this starting sort of stage. And it's the same thing that happened in mitosis. You start with a chromosome, and in this case we're going to look at homologous chromosomes, which is what the blue and the red mean. So we've got blue, let's say this is paternal, i.e. from your dad, the red being maternal, being from your mum, and these are a homologous pair, which means that on this piece of chromosome, which is made up of DNA, a certain bit of that DNA will code for a gene of some kind. Now let's say this is eye colour. It's not as easy as that in the real life, but that's what we're going to use. If we look across to the corresponding pair, the homologous pair, which has come from your mother, it will have the same genes, i.e. it will have a code for eye colour. And that's what's meant by a homologous pair. Homo means same, logos means position. It means the genes are in the same position. So we've got an eye colour here and an eye colour over here. Now the fundamental difference is that the eye colour over here might be blue, being passed down from your dad, and the eye colour over here might be passed down from your mum, which is green. They both code for the same gene, which is eye colour. However, the allele is different, i.e. the expression is different. So one will code for blue, one will code for green. Okay. Now, in terms of the numbers, it's very important in the same the same process as what happened in meiosis, is to start with the chromosome makes an exact copy, and if you can remember, we call that a sister chromosome. And you can see this is what happened. This is what has already happened here. You can see it's almost in an X shape. This is one chromosome which made it an exact copy, and this is one chromosome which made an exact copy. Okay. Now, in terms of the numbers, you have 23 blue, 23 red. Even though there's a sister attached, if you can remember, by a centromere in the middle, the X, the sort of copy and itself is classed as one chromosome. So we've still got 46 chromosomes to start with. However, they have a sister each attached, which explains everything as to how these numbers come into action. Because when this first split happens, you can see that in meiosis 1, this chromosome goes this way and this chromosome goes that way, which leads to this first split up here. And if you can look at the diagram, so 46, which was up here, goes into 23 and 23. That's the first sort of issue in terms of numbers. Now, when we initially said, or the key question is, how does a cell with 23 chromosomes split into two cells with 23 chromosomes? What's going on is these sister chromatids now are splitting. So we get that split and that split. So essentially we get this, we get this, we get this, and we get this. When they're joined together as sisters, they're only classed as one chromosome. When they split apart from their sisters, that's when they become their own chromosome, and that is how we can create two different cells of 23 from one cell of 23. Now that's the numbers involved, and again, if you didn't follow that, rewind it and make sure you get the numbers involved, because to summarize, every chromosome makes a copy, and even when it has a sister attached, that sister, they're still one chromosome. So we have 23 blues, 23 reds, and when they split, 23 goes this way, 23 goes this way. So we'd have 23 there and 23 there. And when the sisters then break off, they become their own chromosomes in their own right. And that's where we create, from 23, we get two 23s down here and 223s down here. So that's the numbers involved. So that's the first aspect that you need to try and get across in your answer for the test. The second thing you need to get across is how we've gone from a cell of 46 chromosomes, as in any of your cells, and we've created these 4 cells here down at the bottom, which could be sperm or egg. And if you look at them, they all have different genetic makeup. So this one is purely being passed down from the dad. This one's mainly dad, but a little bit of mum. This one's mainly mum, but a bit of dad. And this one's all dad. All mum, should I say. Did I say that right? Hopefully I did. All dad, mostly dad with a bit of mum. All mum with a bit of dad, and all mum. Yeah. So how did it get to be like that? Now, if any one of these sperms or eggs fertilize with somebody else as sperm or egg, then that's where we get a completely new set of genes. And that's where we get a completely new person with different characteristics. Unfortunately, that is why you do not look all like your dad or all like your mum. You'll see that these genes are being passed on through your generations. Now, to keep it simple, like I tried to say at the start of the video, your 2 key points are right here. Chromosomes line up in homologous pairs along the metaphase plate. They randomly align to either side, creating genetic variants. So it's important that we look back at mitosis, and let's say if we had a homologous pair, which is what we've got at the top here, rather than lining up side by side, this is in mitosis, you'd get them lined up on top of each other, so that when they split, they would get an exact copy of each side. You'd get the chromosome from dad going this way, the chromosome from mum going this way. You get the chromosome from dad going this way, you get the chromosome from mum going this way. And in that way, you make an exact copy. Now, here is the fundamental difference between mitosis and meiosis. In meiosis, the chromosomes line up in homologous pairs along the metaphase plate. Remember, metaphase means meet. So you can see that this pair have found each other and they line up side by side. The difference is that although the maternal chromosome is on this side and the paternal chromosome is on this side, it is completely random which side they go. So chromosome number 1 might look like this, but homologous pair number 2 may well have the maternal chromosome on this side and the paternal chromosome on this side, and it's completely random how that happens. That is the first key point that you need to try and explain, which is summarized in this point. Chromosomes line up in homologous pairs. They randomly align to either side, creating genetic variants, because what happens is that when we have this first split, i.e. meiosis 1, you get so much of mum and so much of dad going in different directions. You might have 3 chromosomes that of dads mixed with 20 chromosomes of mum, and you'd have the opposite on the other sides. That is the first way in which we get genetic variants. Now, if you've got any extra questions to that, and if you rewind it and it doesn't make sense, then please shout up and come and ask me. Because the second and key reason why we get genetic variants, you may have seen or may have noticed that some of these chromosomes, although mainly blue, have a little bit of the opposite colour on, and the reason why is it relates to how they line up in this metaphase again. You can see that when these 2 homologous chromosomes form the tetrad, which is when they come together, they overlap, so their tails can overlap. Now, this doesn't happen all the time. Again, it's random. But when they're overlapping, these chromosomes here, when they move apart, when they're ripped apart by the the spindle and the kinetochores, etc., then a bit of mum can actually come off with the majority of dad, and vice versa. But the beauty of it is that the genes here are the same genes. So let's say this is for shoe size. So this shoe size here is mum's, and her shoe size was 8, let's say. Although obviously the genes don't code exactly for shoe size, foot size, but I'm just using it as an example. And the shoe size over here from dad was a size 12, big feet. So what we've got there is a chromosome that is mainly genes from mum. So she might have blonde hair, she might be 3 foot tall, she might have, I don't know, a specific shaped nose, but she may well have had the shoe size from the parent, or from the paternal chromosome, which was a shoe size 12. Although that is a very funny picture, a 3 foot tall blonde lady with size 12 feet. But anyway, that's how it can go. And this is called breaking off. This is called breaking off, or crossing over. That's also another term for it, where the chromosomes cross over one another and they snap off when they break apart. But it explains how we get genetic variance, because although you might on one chromosome have the majority of dad's chromosome, you may well have some of mum's too, in a minority. So, to cut a long story short, in summary, there are 2 things that you need to explain really. There's the numbers. They line up in homologous chromosomes, they split, and then the sisters split. Just wait for you to take that in a second. They line up in homologous chromosomes, each with a sister attached. In meiosis 1, the homologous chromosomes split. In meiosis 2, the sister chromatids split. That's the numbers. And in terms of the genetic variance, they randomly align to start with. The homologous pairs randomly align, which means there's a different amount of red and blue on either side. That gives us genetic variance, because when they split, there's different red and blue on either side. And then when the first split happens, parts of chromosome break off either chromosome, and so you get a different genetic makeup when you come down to the second split. And that is, or they are, the key chromosome movements within meiosis. I hope that was useful. Again, it's never going to go in straight away. Pause it, absorb it, learn it, move on. If you've got any questions, come and see me, because we need to get you well prepared for that test.